In the Antioquia region of Colombia, there is an extended family carrying a mutation known as PSN1E280A. For decades, the medical timeline for those born with this genotype has been starkly predictable. A carrier typically reaches mild cognitive impairment by 44 and progresses to dementia by 49. In 2023, researchers identified a man who deviated from that path by 3 decades. He remained cognitively intact until 67, resisting a fully penetrant dementia 23 years beyond the median. Genetic analysis located the driver of this resistance, a single heterozygous amino acid substitution, histidine to arginine, in the gene encoding the protein reelin. Nature essentially ran the experiment first: 1 substitution, 1 lifetime, and a clear clinical readout. The task for neuropharmacology is no longer to hypothesize if a target might work, but to determine if we can reproduce the effect of a mutation that already has. The autopsy of this carrier clarified exactly what the protection looked like at the level of the tissue. His cortical amyloid burden was extreme, higher in fact than that of younger, already impaired carriers in the same kindred. While amyloid spread unchecked, his tau pathology was strikingly limited. In the entorhinal cortex, where Alzheimer's ordinarily begins, the tau signal remained low. Knock-in mouse models carrying the variant mirrored this result. They showed stronger activation of reelin's canonical adapter, disabled 1, which in turn reduced the phosphorylation of human tau. This causal chain, from reelin to disabled 1 to reduced tau, presents a specific design goal. Because the variant increases signaling, it's considered a gain-of-function mutation, implying a drug should simply aim for a better binding version of the reelin protein. In this instance, a cognitive reprieve was achieved without reducing amyloid. This suggests the therapeutic objective is not to clear plaques, but to replicate the localized, reelin-driven brake on tau. Reelin is a massive glycoprotein, 3,460 amino acids long. Its architecture consists of 8 tandem reelin repeats, followed by a short, highly basic C-terminal tail. The receptor-binding activity resides entirely in a central fragment from repeats 3 through 6. This region is both necessary and sufficient for gripping the receptors ApoER2 and VLDLR. However, the COLBOS variant is not located in that fragment. Residue 3447 sits inside the C-terminal tail, 783 residues beyond the end of the receptor-binding module. Cell-free binding experiments confirmed the significance of this distance. The COLBOS substitution does not alter reelin's binding to either ApoER2 or VLDLR. This means the variant's protective mechanism is not a matter of receptor affinity. Whatever bought this man 23 years of cognition, it did not work by improving reelin's direct contact with its primary receptors. The tail functions instead as a staging ground. It interacts with membrane co-receptors, such as neuropilin-1 and heparan sulfate, which help anchor the larger protein to the neuronal surface. The COLBOS variant specifically improves reelin's grip on these co-receptors, fine-tuning the interaction rather than dramatically increasing it. This matters because of how the signal is fired. A monomeric fragment binding a single receptor is virtually inert. To activate the pathway, reelin must cluster multiple receptors together simultaneously. By holding the co-receptor bed more effectively, the variant increases the probability that receptors will align on a single patch of membrane.
It forces a cluster, shifting the gain from affinity to avidity. The variant's true advantage is in residency, making it more likely that enough individual receptors close together at once for the signal to fire. If the variant is so effective, the immediate impulse is to deliver the mutated protein or its gene directly to a patient. But this approach is blocked by 4 distinct constraints. First, the protein is too large. Reelin is 388 kilodaltons, while current blood-brain barrier shuttles are engineered for antibodies at less than half that mass. Second, the gene will not fit in a standard viral vector. The coding sequence is over 10 kilobases, while adeno-associated virus has a limit of 4.7. Oversized genomes are truncated during packaging, not compressed. Third, delivering a smaller fragment of the protein fails to solve the problem. The only fragment small enough to be vectored is the central module, which by definition lacks the tail containing the mutation. Finally, while the single nucleotide change is a textbook target for base editors, current in vivo base editing is restricted to the liver. There is no established method for editing these residues in the central nervous system. A literal phenocopy is foreclosed by the limits of delivery. A drug cannot copy the molecule itself. It must copy the architecture the molecule creates. If the therapeutic trigger is receptor clustering, we do not need to deliver reelin at all. We can bypass the massive ligand entirely. Research from 2004 shows that bivalent agents, like engineered antibodies, directed at the receptor's binding domain, are sufficient to induce phosphorylation and increase synaptic potentiation. A bivalent antibody achieves directly what the variant does indirectly. It forces the receptors together, becoming the clustering agent itself. This leads to a counterintuitive design rule. The drug that best imitates this reelin variant is the one that looks least like reelin. This strategy must navigate a significant collision between two independent areas of Alzheimer's research. Pathological tau enters neurons by binding to heparan sulfate proteoglycans on the cell surface. To stop tau, a common strategy strips this sugar bed away using enzymes or knockouts. However, reelin uses that exact same sugar to stage its break on tau. Stripping the sugar simultaneously silences reelin signaling. The COLBOS variant makes this paradox even more dangerous. Because the variant depends on heparan sulfate twice as much as the wild-type protein, it is more vulnerable to these interventions. A drug designed to strip the sugar would actively work against the brain's endogenous protective mechanism. This collision defines a strategic constraint for this therapeutic space. An agent must target the protein that reads the sugar, never the sugar bed itself. This approach, targeting the protein side of the interface, has already been validated in vivo by the mirror program for the Christchurch variant. It allows the protective signaling to remain intact while the pathology is addressed. The goal is not a maximal activation of the pathway, nor is it the clearance of amyloid. The true goal is a modest, sustained clustering of receptors at the cell surface, designed specifically to maintain a chronic, long-term break on tau. We cannot build a reelin double, but by understanding the biophysics of avidity, we can build an understudy, a molecule that hits the same marks even if it doesn't look the same.